A comprehensive procedure to evaluate the interplay between the complement protein C1q and hyaluronic acid in promoting cell adhesion.
We have recently defined that the complement component C1q, a molecule of the innate immune system, is highly expressed in the tumor and placental microenvironment and is able to interact with component of the extracellular matrix, in this case the hyaluronic acid.
We propose a method for studying the biological functions of proteins present in the stroma microenvironment, in order to investigate the ability of these molecules to shape the cell adhesion properties of extracellular matrix factors.
In order to study how this interaction can be censored by the cells present in the tissue microenvironment, we set up this easy method in which primary cells derived from human tissues were allowed to adhere to C1q bound to extracellular matrix, in this case to hyaluronic acid.
For coating procedure, the first step is the binding of high molecular weight hyaluronic acid to 96 well tissue culture treated microplate. For coating, hyaluronic acid is diluted in 0.1 mole carbonate buffer, pH 9.6, at a concentration of 50 micrograms per milliliter, and gently pipetting is required to achieve a homogeneous solution. 100 microliters of the solution are used to coat the wells. The plate is then transferred at 4 degrees Celsius overnight. Next day, vacuum aspirate the treated wells. Wash once with DPBS. Add either C1q or BSA in the corresponding wells. C1q is nowadays